oh, it's safe to invest because it's a good product.
What do you think have been the greatest achievements or failures within the last 4 decades, and why do you think they have or have not worked?
The biggest failures are to do with, I think they can all come back to one thing, is we do not understand fish. And we don't understand fish as an organism, and we don't understand fish as a domesticated species. So they're two slightly separate questions. And if you actually move into agriculture in a more general sense, we have domesticated plants and we have domesticated animals. Mm-hmm. Now, if I ask you, how long ago did we first domesticate our plants?
That's like 10,000 years.
So 10,000 years. How long ago did we first domesticate animals? About 5,000 or 6,000 years. So we've got 5,000 or 6,000 to 10,000 years of empirical experience. Did we know what we were doing? Not really, And it wasn't really until Mendel came along that we had this advent of genetics and, but people would breed things and something would come out. So and that's what I mean by empirical.
Yeah.
Today we understand plant breeding in a way, way different way altogether. Mm-hmm. And the scope of some of the experiments that go on to look for particular varieties. I'm a president of a society of experimental biologists, and so I see botanists talk about this, and crop production. It's phenomenal what's possible now. And so that is one thing of understanding that side of domestication. And now the industry, with a good solid financial base, which means sales, is now starting to look at that level. Which varieties are the best? Mm-hmm. Which varieties have more innate disease resistance that you don't have to treat them with certain things. Then there's the vaccinations. And so vaccinations are equivalent to vaccinations in animals, and they'd be equivalent to treating with herbicides or pesticides or something like that with plants. And yes, minimize them, and if you can find something that is tolerant. So that's the one side. And the other side is fish breathe water, not air, and that's fundamentally different. And they use their breathing organ as an excretory organ too. Life in fresh water and salt water are totally different challenges physiological animals, and so that's the other side of it. And I think that understanding disease resistance in these animals, understanding the nutritional requirements have all been major step forwards that have come. And as a result, I think we've crossed significant hurdles.
I have a question. Last year I saw a presentation by someone that was speaking on GE salmon, and so that seems to be genetically engineered salmon, and so that's a progression in, say, like domestication of plants, and now we're seeing that research in fish. What are your thoughts on that?
I think people can be concerned about what they eat and should be concerned about what they eat. What I do know is, because I happen to give lectures on the digestive process sometimes, is that we take a very complex food substance and then we chop it up into little bits so it can move across a membrane. And so the little bits are then reassembled inside of us to useful products, and sometimes if we carry toxicants then they're not so useful. So genetic engineering represents different proteins, but primarily a different DNA sequence. Mm-hmm. And so what we're asking is, if we eat something with a slightly altered genetic structure to the original wild variety, should we be worried about it? And I would say, well, if you're worried about a slightly altered one, why eat anything at all? Because it has a different DNA. Mm-hmm. And so I think from the eating point of view, we should not be fearful of it, and I think the public needs to hear that simple statement. If you're worried about the spread of GMO, then I think that's a different thing altogether. And I think that sterility has to be an integral component of any GMO. And if you actually look at the work that's been done on GMO of salmon, it's that they produce sterile. And
Yeah,
so,
I saw that.
and so one of the big things is how do you make sterile fish? And it turns out that the pioneers of making sterile fish were also in British Columbia.
Yeah, actually, there's quite a research station just here in West Van.
Yeah, and UBC used to share part of that building. We used to do research. I still have an office and a lab, and I do experiments there, courtesy of the Canadian government now.
Thank you.
Do you have any issues with promoting or funding with your research with the salmon industry?
I have no problem being funded by the salmon industry for research. I do research where I know I have expertise and can do high quality research. How do I measure high quality research? In the past, I just did I had to do a grant renewal, so I had to do some bean counting. So in the last 6 years, I know that I've had 3 papers in Science, a letter in Science, a letter in Nature Communication, paper in Nature Communications, a letter in PNAS, 3 papers in the Proceedings of the Royal Society of London. And that's just part of 123 publications in the past 3 years, 7 books. So I do write a lot. I tell them I'm only interested if you allow me to publish the results. So in other words, the results are the results, not any preconceived ideas that I may have in the way of a hypothesis or that you would like over some other. And I have never in my life had an industry say no. So they've always said, publish the results, and I've always published the results, and the results have said what we've discovered. And so that's, I think if you can say high quality, people will come to you and look for that.
Where would you say this desire for transparency in this industry comes from for you? Is that because it seems like you wanting to republish the results? Is that as a response?
For me, so the transparency for me is that science is about quantifying something and then communicating that information for the betterment of the world. The basic research model is you have a pyramid, lots of basic information, and then it filters up, if you like, to be applied. The worst thing we can do in this world is flip that pyramid upside down. So the driver is the applied work, and then you sort of say, well, what basic stuff do we need? And so I think that my questions are always start at some basic research and then they feed into the applied. And so you have to be transparent, because basic research is transparent in the sense that you're meant to publish it and you're meant to communicate it. I made a concerted effort to you may ask me what fish physiology is, but I'm not going to even try. It's how they work.
Yes.
And so I decided that I'd spent my whole life in fish physiology, and nobody really knows what it is, let alone pronounce it sometimes. and so I produced an encyclopedia. And so I got a whole bunch of my friends and colleagues, some people I knew, some people I didn't know. And I said, I want to take a topic and I want you to make it as simple as possible that a high school teacher could use it. So in other words, not dumb it down, but make it more accessible. And so my fundamental belief is that you communicate, that's one. And my second one is that all applied projects should stem from some basic theoretical concept. And so those are the 2 tenets that I hold very closely to my research.
Where do you see your work and research going in the future?
Into retirement. I actually think that that's for others to determine. Some people don't like what I have to say, and that's their view of the world. It doesn't worry me. It's because I have the tenets I have. It doesn't change anything. It's just that you don't like what I have to say. You know, if I tell you that the footprint of UBC is more than all of the sea cages in British Columbia, you can believe me or not believe me, but it's a fact. They could all fit into Stanley Park. Would somebody say that UBC is a big footprint in the whole of the Lower Mainland? Some people say yes, some people say no. Would some people say it's a necessary evil? You know, is Stanley Park very big? Well, if you'd have tried to park yesterday at the aquarium, which I tried to do to take my 2-year-old grandson from Australia to go show him some BC fishes, it was impossible. So it's not big enough. There's not enough parking. You know, so we're expanding the aquarium and there's not enough parking. And the aquarium's a great thing for showing. I mean, they asked at the Marine Mammal show, how many people have seen white-sided dolphins in the wild? So everybody was around there. There's only one hand that went up. It was my wife's. We can see the wild stuff here, and we can. But the true education is that most people don't see that, and so we have to do it in a responsible way. And I think that's the job of scientists, to be responsible. So I'll be more responsible, more open to interviews, and I hope this helps you.
Absolutely.
Yeah.
Do you have, as a closing question, do you have any hopes, expectations, or concerns for the salmon industry in the future?
My hope is that they continue in an open, transparent way. My hope for you is you invite people from the BC salmon farmers down here to actually be part of your interview process, because I think you'll see that they're trying to be transparent. And my really biggest hope is that people pay more attention to what I would call facts rather than factoids. Unfortunately, you know, there's 3 cell phones sitting out here, and that's part of the breakdown, is that you're not going to get anybody listening to me for half an hour. And that's why I asked you one question. I only asked you one question. What was that?
How is this information going to be used?
That's right. Because I know that you cannot have me talking. As interesting as you are. Anyway, so that's my worry, is that dilution can lead to misinformation. The devil is always in the... You can't even complete it, can you? The devil is always in the...
No.
Detail.
Detail. Okay.
So at some point, there's a big message, simple message, a sound bite, but underlying it is some detail. Without the detail, you're stuck. Okay?
Thank you.
Thanks.
Good, thank you.